we also, at that point, need to make sure those proteins stay folded. So in our cells, we'll have other proteins that ensures the proper folding of that protein, which is crazy. So let's take a look. So this is kind of talk about it in a different method. Right here, about the different levels, primary, secondary, tertiary, how we get those. Now, right, just a few more minutes. Protein denaturation. Some proteins can unfold. That is what we call protein denaturation. Remember, protein denaturation is different than breaking apart the protein. In order to break apart the protein, you need enzymes. So then you can free up an amino acids. That's not what this is. Protein denaturation means that you have somehow unfolded the protein. Right? Now, in our body, we need to unfold it before we're able to have our enzymes, right, in our stomach, in our small intestines, before the enzymes can work. Protein denaturing means that you have this small, now these proteins that you've just eaten, they're very, very tightly folded together. If they're tightly folded together, how are enzymes going to get in there and break it apart? It can't, right? It's too tightly folded together. So what happens? You need to unroll it, right? You need to unroll it first, and with that unrolling and unfolding the protein is called denaturation. We see this all the time. Big example is this. When you cook and you're eating eggs, not the egg yolk, but the egg white. What happens with the egg white when you cook it? It gets fluffy, right? It used to be solid and it should be kind of, clear, right? But once you start cooking it, it turns fluffy white. Why? What we're doing when we're cooking it is putting a lot of heat in there. Heat naturally unfolds a protein. So as you introduce heat, all the proteins found in the white of the eggs, all those proteins unfold. As they unfold, they denature, they become fluffy. Have I broken the bonds? No. All I've done is unfold it. That's it. Now the crazy thing is that it can happen to us. I don't know, a few years ago, if you, now it's like, it's a long time ago. All right, I was in medical school, and that's like 15 years ago. Anybody's ever heard of a football player that died from heat exhaustion? His name was Corey Stringer, and he used to play for the Minnesota Vikings. And what happened was he was, older, right? I mean, older relatively for football. He was like 32, right? So he's not old, but older for football. He's a big guy, 6'6", 370 pounds. He's a big man. What happened? Well, what happened to him was a starting left tackle for the Minnesota Vikings. So in football, you practice in the heat. So they practiced in the heat, and he had heat exhaustion one day. And instead of letting him rest, he went back and went to practice the next day. Just as hard, just as hot, right? His body temperature, when he came in and he passed out, his body temperature was 107.5 degrees. Right? That's how hot his body temperature was. What happened? Well, at that 107, 108 degrees, the proteins in your brain start to denature. Right? As they start to denature, the proteins there that helps it reshape also are lost and denature. Right? And what happened? He went to a comatose state immediately, had seizures, and he died as a result of this. Right? So high fevers can cause denaturation of the protein in the brains. That's why some people have seizures as a result of this. If we can decrease the fever, then we can kind of lower the body temperature, and then hopefully the proteins can be reshapen. But there's no kind of foolproof way of doing that. Yes?
So it's not too good to stand, well, I know it's not too good to stand and eat too long.
Yeah.
But it's not
The
good.
thing is this, right? I don't understand how in Florida, in the South, they can play football in that heat, man. Right? You see them wearing full pads, and it's like 100 degrees, and the humidity, is like 85 degrees. I remember the first time I went to Florida, I'm like, how in the world is it, 85% humidity, 90% humidity, and it's not raining, right? So it's hot, it's humid, and now you have all those pads on. Usually what happens is they have a lot of breaks, right? They have a lot of water breaks. But you guys, if you played football early on, when I played,
But if you're
right?
born there, your body would adapt to it, right?
No,
In Florida.
no, your body ain't adapting to it, right? Your body is not gonna adapt to that. So what happens is, and that's one of the reasons why people die, is because you think your body adapts to it. No, it's gonna keep causing you to sweat. What happens if you keep sweating? You start losing more water. What happens if you lose more water? It makes it easier to increase your body temperature quickly, right? And you're exercising, which increases your body temperature already. So you have this bad positive feedback loop that if you don't break, you're gonna die. Nowadays, I think they are mandatory if it's after a certain temperature that they have to cancel practice, which is good, right? But yeah, there's not much you can do. I think the highest that somebody has ever survived the fever, very luckily, and not have permanent deficit was like 114 degrees. Anything over 105, 106, I'm freaking out, right? I'm gonna make sure at that point, we'll even do, kind of cold water baths, right? Nothing is beyond kind of an option at that point, right? Tylenol, Motrin, right? Like constant every 4 to 6 hours. Yes?
So if somebody have a fever, should you pour cold water on them?
No. I said if you have a high fever. We're gonna see, right? Most doctors will tell you, do not do anything unless the fever is above 100 or 101 degrees. Certain doctors have certain kind of barriers that you have to break, right? The thing about fevers is this: when you have a virus, usually your body's causing the fever, right? Our body's immune system is incredibly good, right? It's great at a body temperature that's slightly normal to high. 100, that's about the top end, right? Now, there are some viruses that makes it spike. Swine flu increases your body temperature over 102 degrees. Most of the time when a doctor tells you, don't call me unless it's 101 or 102, is for one specific reason, right? After 101, 102 degrees, it's more likely to be a bacterial. And now I can prescribe antibiotics. Under 101 is most likely gonna be a viral. And if it's a virus, it's your own immune system increasing the body temperature. So they're not, they're not gonna prescribe anything. It's its own body doing it. Now, how come our body doesn't then stay at 100 degrees then? Right? If our immune system is more efficient there, why doesn't it stay at 100 degrees versus 98.6? Simple. Have you ever had a body temperature of fever that's only 100 degrees? You feel terrible, right? Even when it's slightly higher, you feel tired, right? You feel lethargic. All you want to do is sleep. That's when it just got to like 100 degrees. So we don't like the body temperature that high. It only does that when it has to. All right, now, enzymes. The only thing I'll ask you about enzymes is because you'll see enzymes, right, in our cells and in our body. Usually enzyme has an kind of a suffix ase in it, lactase, sucrase, right? Things that allow you to break things apart. Now we're going to talk more about enzymes in 202 when we talk about how the actual GI tract breaks apart the materials we eat. How do we digest it? Right? And again, this is the binding site that I want you to look at. Very specific. So even though glucose and galactose look almost identical, right? They look almost identical. The only difference here between glucose and galactose, they have the same chemical formula. So, here's glucose. Here's galactose. What's the difference? Just this carbon. The oxygen is facing one direction in glucose. The oxygen is facing the other direction in galactose. That's the only difference, right? The weird thing is this. A glucose receptor will not bind with the galactose receptor. That's how specific these proteins are. A galactose receptor will not bind with a glucose receptor. Just can't do it, right? Even though the only difference is this carbon. That's it. But that's enough, so then our binding sites right here are different. So here's a binding site, and how are they formed? By folds, by folding, right? Primary, secondary, tertiary structures allow you to form folds that then allows us to form the specific binding site. All right, so if we only have just a small microscopic change between glucose and galactose, our binding sites are so specific that if it's glucose and you have a galactose molecule or a galactose receptor, right, it's so specific it won't even bind to it, much less bring it in, right? So the protein shape, it makes the binding site, and the binding site is incredibly specific. Right. Now, last kind of macromolecule is DNA and RNA, or nucleic acids. Now, next week we'll start chapter 3 and discuss like all the different types of nucleic acids, stuff like this. Now, I'm going to discuss it first here, and then we'll end. Nucleic acids are, there's two main types, right? Most of you have heard of DNA, deoxyribonucleic acid, right? That's the genetic material that we see inside the nucleus, nuclear DNA. It's a genetic material that is passed on from our parents to us. DNA is a combination of a sugar, right? The sugar is called deoxyribose. DNA, deoxyribonucleic acid. So the sugar, right, has one carbon that doesn't have oxygen. Deoxy, missing oxygen, right? So deoxyribose, right? So it has a sugar. It's got a base right here. Sometimes you might have seen certain movies where they just, all they do is show a strand of DNA, and you do see that. I remember watching it in Jurassic Park, right? In Jurassic World. You see a strand of DNA like this, a double helical strand, and then all you see on one side is an A, or a T, or a G, or a C, right? Those are what we call the base, nitrogen base that sticks out of our nucleic acid, right? That's what gives our nucleic acid its specificity. All nucleic acids have this core of a sugar and phosphate. What makes one nucleic acid different from the next is the base that sticks out of it. There's 4 main bases. There is adenine, thymine, guanine, and cytosine. 4 bases right here, right? Those 4 bases are part of a double strand that allows us to face inwards right here. So if you see a thymine base right here, it faces inside. The sugar and the phosphate of DNA forms the backbone, right? Now, the backbone means that it forms that main structure, keeping everything together. So in DNA, you have double strands, like we see here. Who's ever been to Discovery World? If you've been to Discovery World, right, when you go in, I believe to your left or right, when you walk in, there's like this, kind of structure where all those little wood things are. You press a button and the wood moves, right? But if you go up and you look at the staircase intertwines, double helix. It's supposed to be DNA, all right? So next time you go to Discovery World, look at the staircase. You will notice that they kind of twirl like this. And if you look in the middle, you'll also notice that there's something that expands and moves back and forth. Those are the bases that's supposed to be sticking out. So what we see in DNA are these letters, the bases that faces each other, each strand. The actual material that forms the double helix, that's made out of ribose or deoxyribose, and phosphate. Ribose, deoxyribose, and phosphate. It forms that backbone, right? It forms the larger structure, right here. Now, the reason why I'm mentioning it is simple, right? You guys have heard of DNA type of, DNA exams, DNA tests, right? For paternal, right, to see who's the dad, DNA tests to see your normal, most people do nowadays do 23andMe, right? What happens is, it's great until you find out that you're not part of your family, right? There have been kind of reports where somebody buys your parents a DNA kit, right? And then buys it for the whole family, and one child is not from both parents, right? And they just find out that. So that does happen every now and then. So what occurs here is this, right? We are looking at the sequence of the DNA and seeing if it matches what your parents can give you, right? If your parents can't give it to you, they're not your parents, right? It's kind of sad, but they still love you, right? Hopefully. But they're just both possibly your parents then. So what we're looking for when we do a 23andMe is the presence and the sequence of which of these 4 bases are found in certain regions. DNA is thousands and thousands of these bases. Thousands, right? I would even say, right, more than thousands, right? Billions of these, hundreds of billions of these bases. And you have 46 chromosomes, right? So that's a lot of bases. Usually when we do those DNA tests, they look at certain regions only and compare yours to your parents to see if it matches, right? Now, you might have sometimes heard of PCR, right? And basically what it allows us to do when we do genetic testing, if we're missing one whole side, right here, if all we have is one side right here, that's all we need, right? There's a rule called the law of complementary base pair. What it means is this, right? If we have on one side of our double-stranded DNA, if we have an adenine, then on the other side, we need to have a thymine. Why? There's a reason why you always see A with T and T with A. Why? Simple, right? If you take a look at adenine, it's huge. It's double ring. If you look at thymine, it's small. Perfect. You have one side, a larger base, and the other side, a smaller base. It reacts, makes it nice and uniform. How about guanine and cytosine? G and C are always found together, right? Why? Well, take a look. One's big, one's small. Perfect, right? You want that. Now, you have those 4 bases, adenines with thymine, big and small, right? Guanines with cytosine, big and small. It allows us to have a nice uniform shape. If it was the other way, if you have guanine reacting with adenine, well, they're 2 big bases, so you have a bulge on one part, right? And then it gets small, and then you have a bulge. There's no uniformity, right? By having these 2 bases next to each other all the time, it gives us the ability to have a nice, straight, uniform double helix that we see in DNA. Now, adenine's always with thymine, also because it maximizes the hydrogen bonds. By the way, this hydrogen bonds is the only thing keeping your double-stranded DNA together, right? If we break apart the hydrogen bonds, your double-stranded DNA falls apart. They will no longer be reacting with each other. Now, remember what I said. In the laws of complementary base pair, if I know in the sequence of DNA, if I know one of the bases, I know them all. So right here, right? I know for every adenine, there's a thymine. For every thymine, an adenine. For every guanine, cytosine, right? By knowing one side, you know the other. You don't need both sides. Right? We know that. What else? If we know in one region, let's say this region right here, I tell you, that region has 17 6.5% of that region, it has cytosine. By knowing that one number, I should be able to tell how much guanine, how much adenine, and how much thymine. So if I ask you, right, this region of DNA has 17%, 17.5% cytosine. How much thymine do we have? And it's not 17.5%, right? How can we tell? Well, the cytosine and the guanine are found in equal amounts, 17.5%. Oops, all right, between those two, we have 35% of our DNA. All right, that means these other two make up the other 65%. And they're found in equal numbers. 32.5%, 32.5%. Combine, you add it all up, 100%. Because all those bases together should add up to 100%. If I know one number, I know them all. All right? This is the law of complementary base pairing. Now, we'll talk more about that next week as we kind of go into more depth. Right? Next week, when we start talking about how we make DNA and how we make proteins, right, then it becomes kind of a little bit more challenging. Right? So make sure you read all of chapter 2. Right? It's up to you. Right? We can kind of describe it, right? And it might make sense as we go through it, but you need to also read it when you get home. Because if you don't reinforce what you just learned. And if you don't reinforce it right away, you're just kind of starting from the very beginning again, right?